All right, thanks for the opportunity to speak. So I'm going to talk today about a machine learning approach that we use to identify parts of the genome associated with vocal learning behavior. So first, I'll give a quick introduction to how vocal learning evolved, talk about how we identified vocal learning genes, and then go into some of the new biology in our method. So first of all, I wanted to mention that vocal learning is an example of convergent evolution. Like a marine behavior where you can see physical similarities in sharks and marine mammals that have evolved in different lineages. And so vocal learning is also an example of convergent evolution in that the ability to mimic sounds has evolved in different lineages. And it, of course, forms the basis of human speech. So humans have the ability, bats, whales, and dolphins, songbirds, parrots, and even some hummingbirds have the ability too. And actually, if you comprehensively look across the mammalian and avian evolutionary tree, you'll see different instances of vocal learning evolution in these branches. Now, it's also notable that non-human primates do not have this ability. And so we have all of these species with vocal learning ability and without vocal learning ability. And so that forms the foundation for a comparative approach. All right, now there are different convergence at multiple levels. I talked about behavior, but there's also neuroanatomical convergence. And I want to highlight two neural circuits that vocal learners like songbirds and humans have that closely related vocal non-learners don't. So there's a loop from the cortex or the analogous region of the cortex pallium to the striatum and back up to the cortex that's found in the vocal regions of songbirds and humans, but not the chicken and macaque, and also direct projection from the motor areas down to the hindbrain here that isn't found in the vocal regions of species like chicken and macaque without vocal learning ability. Now, some of the research that I did in the past as a part of the Jarvis lab was to identify cases of molecular evolution. And so what we did is compare the vocal region RA to the nearby region, the motor archipallium or analogous regions that participate in other motor behaviors. And we actually found certain genes that were specialized for vocal learning across finch, budgerger, hummingbird that weren't specialized in dove and quail. And notably, a lot of these genes also show molecular specializations in the part of the human motor cortex that specialize for the production of learned vocalizations, including SLIT1 here, which you can see is lower in the vocal motor areas. Now, with that in mind, we wanted to focus on the genetic level. So basically identify genetic loci that are associated with vocal learning across mammals. And so we first wanted to look at the level of genes. So I showed this diagram before, but basically we had all of these species, cetaceans, pinnipeds, and bats with vocal learning ability, and of course humans. And so we did some protein comparisons. So we used the multiple genome sequence alignment from the Zoonomia consortium that we're a part of for 471 proteins. We fed these into RER Converge, which is a package for looking at relative evolutionary rates of proteins. And actually, we did find numerous proteins associated with vocal learning. You can see the p-value distribution shifted to the left, suggesting that we've identified several significant candidates. Now, overall, we found 201. And I want to highlight two of them that were highly significant, TSHZ3 or T-shirt3, but also ZNF536. Both of them zinc fingers and both of them transcription factors. And transcription factors were enriched in this list of 201 proteins. But notably, these two proteins are actually at the same locus, ZNF536 and TSHZ3 here. And actually, TSHZ3, when it's deleted within the human population, is associated with a form of autism that has severe speech delay. And in mouse models, it can actually influence the connection between the cortex and the striatum, a feature of the vocal learning neural circuit. But because of these transcription factor candidates, we wanted to know if there were also gene regulatory effects, as there are well-documented cases where basically differences in gene regulation can relate to complex traits as well, not just differences in protein sequence. But to do that, we actually needed to adapt our methods. So we decided to design a new machine learning approach for identifying vocal learning associated regulatory elements. So we first started by collecting open chromatin data sets from the cortex and also the liver. We even collected new bat tissue with collaborators Michael Yartsev. And basically, we found that regulatory elements are highly tissue specific. And so we could use this as a foundation to identify convergent evolution in regulatory elements. Now, there's an assumption that if something is evolutionarily conserved, it's functional. And you can see that if you look across protein coding genes that at even base pair resolution, you can see highly conserved regions related to protein coding sequence. Unfortunately, for regulatory elements, this nucleotide level conservation is more of the exception than the rule. So one example of this is the scaper regulatory element that regulates the islet gene. And even though this regulatory element has its function conserved between sponge, zebrafish, human, and mouse in terms of driving similar cell type specific enhancer activity, there actually is very little sequence conservation. But what did happen is that basically the same combination, or very similar combination of transcription factor binding site motifs are there. They've just reshuffled to maintain their function. And this is across about 700 million years of independent evolution. So to basically capture this regulatory evolution without conservation of individual nucleotides, we train machine learning models to predict open chromatin in both brain tissue and liver. And then we fed orthologous sequences across species, the 222 Boreoeutheria genomes of the Xenomia project, into these machine learning models to make predictions about motor cortex regulatory element activity. So another way to visualize what we did is we start off with this matrix of open chromatin regions along the rows and species along the columns. We start off with information from a small number of species and then make machine learning inferences to fill in this entire matrix so that we get predicted open chromatin levels across these 222 species of interest. Now, I don't have time to talk about some notes on methods development, but we're able to use convolutional neural networks, which have a better ability to generalize across species in the support vector machines that we also tried. And so again, we feed these orthologous regulatory elements into the convolutional neural network, and then to find trait-associated loci, vocal learning-associated loci, what we do is we want to find cases where maybe vocal non-learners are predicted to have a high level of open chromatin or regulatory activity, while the vocal learners as a group are predicted to have lower levels. So we do this using the phylolem package to control for the phylogeny. And then we also use another method, phylogenetic permutations, to make sure we don't get false positives. And we come up with some interesting candidates. So for example, we identified a regulatory element that's very close to this VIP gene, and VIP itself has lower expression in the vocal motor areas. And then you can see this VIP regulatory element has lower predicted open chromatin in vocal learners as opposed to vocal non-learners. We actually also found a regulatory element at this locus of TSHZ3 or T-shirt3 and ZNF536, which is also predicted to have lower activity in vocal learners relative to vocal non-learners. And so we can see the ZNF536 and the open chromatin region here and T-shirt3 here. So overall, we're able to get convergence in both regulatory elements and protein coding genes associated with vocal learning in parts of the genome known to influence speech production in the human population. And so this provides a foundation to use mammals as a genetic model for vocal learning behavior. Thanks.